everyone. We don't have a lot of people on today, but this will be recorded. This webinar will be recorded. It's called Lunch with Docs. We have Dr. Peg Nopoulos from the University of Iowa on today with us, and Sarah Jones with PMD Alliance is with us. Margie's in there somewhere too. So, we're going to be talking about the Kids-JHD study at University of Iowa. And but first I'd like to introduce Sarah Jones. She's with PMD Alliance. PMD Alliance has sponsored HD Support and Care Network's webinars and online support groups through the Zoom portal, which has been phenomenal. We've had a lot of people coming on to the support groups and the webinars that have not seen this yet for Huntington's. Huntington's disease has a lot of different programs. There's radio shows now, there's other webinars and live events and things like that, but this is really focused on talking with the doctors, lunch with docs. That's what this is about, so that we can have some quality time focused on talking with the actual doctor. And this is phenomenal to have an audio video program that can accommodate that. So Sarah, tell us a little bit about PMD Alliance.
Thanks, Melissa. Parkinson and Movement Disorder Alliance is an organization that provides support services and education and training for care partners, and also for support group leaders in not just Parkinson's, but in Huntington's and dystonia. And as I was just mentioning, we work with some groups that are with PSP and MSA also. So a variety of different movement disorders. But one thing I would want to say is that if someone's watching this and they either have a Huntington support group or are interested in starting one in their community and just don't know how to get started, regardless of where they're at, but you're interested in starting some kind of a group, you can always reach out to us, and we have a kit that we can send you that can help you get that off the ground, and then we can link you into our quarterly roundtables so that you're linked into a network. So feel free to reach out to Parkinson and Movement Disorder Alliance if you are a support group leader or wanting to lead a group. But I'm excited to hear about this today, and we love our Lunch with Docs.
Tell us real quick also about the InSync for the HD and
yes, great. So we do a 2-day conference for support group leaders, and we cover all of the expenses for people who are volunteer support group leaders, and it has it's mainly for Parkinson's, Huntington's, PSP, and MSA, but it can be for any movement disorder support group. And during the training, we have some movement disorder physicians that speak, as well as people sharing their resources with each other. And we just do some trainings about how to run a support group and how to navigate a group. How do you market it? How do you keep it sustainable? How do you plan for succession? All of those kind of things we do in our support group leader conference, which is called InSync. And we're holding those in California, in Arizona, in Florida, Massachusetts, and Illinois this year. So if you are in those areas or one of the surrounding states, we would love to link you in and let you know when it's going to be.
Fantastic. Thank you so much, Sarah. Okay, so now I'd like to introduce Peg Nopoulos. Dr. Nopoulos is Professor of Psychiatry, Pediatrics, and Neurology, and also Vice Chair for Research Department of Psychiatry at the University of Iowa Hospitals in Iowa City, Iowa. And she is a physician scientist, and her clinical care consists of taking care of patients with Huntington's disease in Iowa's HD Center of Excellence, where she's worked for the past 14 years. Dr. Nopoulos' research focuses on the study of the brain and behavior. This is done using state-of-the-art neuroimaging techniques, specifically magnetic resonance imaging, MRIs. So Dr. Nopoulos is interested in aspects of understanding normal, healthy brains, such as differences in brain structure and function between sexes, as well as understanding how the brain changes with development through adolescence. And she has just been a champion for HD and especially JHD. And so welcome, Dr. Nopoulos, to our webinar. Tell us about what you're doing.
Yeah, so at the University of Iowa, we have a protocol that we've been running for many years. Can you hear me, Melissa? Okay, good. So we developed the program called Kids-JHD about 12 years ago. It actually started in pilot phase. At the time, I was working with the large study called PREDICT, which is one of the largest studies that was ever funded by the government. But that was a study to look at adults who had tested for Huntington's disease. And whether they had the gene or not, they participated in a research study. And it was a brain imaging study. So many of you listening may have participated or know somebody that participated in PREDICT. But what we found in that study is that the brain changes many, many years before the onset of disease. So the onset of Huntington's disease is defined by motor movements. We know that thinking and behavior can happen, problems way before that, but really what we call the onset of disease is motor. And we see changes in the brain a long time before that. And so what we sought out to do was to investigate how far back that goes. So are there signs in childhood that are, representative of the disease that would still be, quiescent, meaning that these, looking in children who are at risk, they have the gene expansion, but they don't, of course, have the disease. They won't develop it for 30 years. Can we see something that is a signature of the illness all the way back there? And so that was the primary, question in developing that program. And then naturally we developed, because we were looking at children, that we wanted to see about children who had already developed the disease. So the Kids-JHD protocol is for children who are at risk. So they have a parent with Huntington's, but they don't have symptoms. They're not manifesting symptoms. And then JHD are the kids who are actually already diagnosed. So not only are they at risk, but they've developed symptoms. They've been diagnosed by a physician, and typically they've had the genetic confirmation for that. So we're going to start in general. I know most people already know about Huntington's and the gene, but I just wanted to go over it just to make sure that everyone was on the same page. And then we'll talk specifically about the programs, the Kids-JHD and the Kids-JHD, what we're looking for, how are we finding it, and who's eligible. So now you can go down 2 slides and we'll talk a little bit about the gene. So
Okay.
this is just a slide, that's a cartoon looking at the basics of, the biology of genes. So the genetic code is DNA. It's made up of what we call nucleotides. You can think of a nucleotide as a word, and it's composed of letters, okay? The DNA code is TACG. Those are the only letters. And then a word is made up of 3 nucleotides. Amino acids are made of a combination of 3 nucleotides, and sometimes those are called triplets because there's 3 of them. So you can see here a picture on the top of a gene, and then you can see that there is, underneath that, there is words made out of the genetic code. And remember, each word is 3 letters long, called a nucleotide. And you can see examples of AAG, CTG, CAG, CGG. The CAG is a repeat. It's a nucleotide, trinucleotide that makes the amino acid glutamine. And then once those molecules get strung together like a string of pearls, you get a protein. And the protein is that multicolored string below. And the protein is actually what does all the work in the cell. So the gene is the blueprint. You put together these amino acids and you make a protein, and then the protein is the thing that drives the function of the cell. So they're all really important. This is the basic biology of how our cells work. Next slide. So the gene that causes Huntington's is called Huntington, and sometimes we shorten that to just HTT. And in HTT, we have a repeat. So in that gene, the trinucleotide CAG is repeated over and over again. CAG, CAG, CAG. And so that means the protein has glutamine, that amino acid. It's repeated over and over again. And importantly, everybody has this gene. Everybody has Huntington's. It's a very important gene. And everybody has repeats. But normally those repeats are only 9 to 25. They're not overly long. They range from 9 to 25. Next slide. And in Huntington's, the repeats are too long. So when the repeats go above 40, we know that that patient will develop Huntington's disease. So another way we name this is that repeats below 40 are gene non-expanded, or GNE, and repeats above 40 are gene expanded. The reason we do that, I know sometimes that, how we name certain things gets confusing. We sometimes use gene positive and gene negative. That makes it sound like you don't have a gene if you're gene negative. Everyone has the gene. So it's one of the ways that we define it is by saying gene expanded, which means that your CAG repeats are too long, or gene not expanded. Okay, next slide. So really importantly, the age of when you get the disease, so we call the age of onset, is determined by how long that repeat is. So this is a graph, it's a figure that shows this relationship. And if you can see at the top, the gray arrow is the age at which you'll develop it. And if you go all the way to the right, you'll see that the CAG repeat number, 36 to 39, we sometimes call that intermediary, or the intermediate zone, because those persons with 36 to 39 repeats, they may or may not develop Huntington's. And typically, if they do, it's later on in life. It's sometimes in the 70s. I've diagnosed patients in the 80s. So if it's in that range, it's later in life. If it's in the range of 40 to 49, if you have 40 to 49 repeats, that's what's called kind of classic adult onset. And in general, the average age there is about 40. And then you can see as we continue to move left, we see that the greater the repeats, the earlier the onset. So if you have more than 50 repeats, you may have an onset between age 20 and 30. If you have more than 60 repeats, you may have an onset before the age of 20. In general, that's considered juvenile onset. It's really important to remember that these numbers are not exact, meaning that there are some patients who have lower numbers who develop it earlier. There are some patients that have higher numbers that don't develop it as early as we anticipated. So there's, of course, some individuality in here, but in general, this is the pattern that the longer the repeat, the earlier the onset. Next slide. So now let's talk a little bit about the programs and who is eligible and what do the kids do. Next slide. So the Kids-JHD program, so subjects are children/young adults who are 6 to 25 years of age. They have a parent or a grandparent. And importantly for this part of the program, they don't have any symptoms. There's no diagnosis of JHD. They're simply at risk. Now, for research purposes only, they provide DNA, either from blood or saliva. And it's important to know that this is a research protocol only, so this is not to get the genetic test. As a matter of fact, everyone is blinded to this. The genetic results are not provided to the family, they're not provided to us, they're blinded. But what we can do is that we can then look at children who are gene non-expanded, so they won't develop Huntington's, and we look at how their brain develops, and we can compare that to children who do have the gene expansion and see how their brain develops. One thing about this arm of the protocol is that this has been going on since about 2006 in its early form. We have recently just closed recruitment into the Kids-JHD. So the kids at risk, we're currently not pulling in any more additional patients at this moment. We hope to open it back up soon, but currently we're on hiatus for recruitment. Now, the Kids-JHD program is very similar. They're subjects who are children, young adults, but most importantly, these are kids who have already started having symptoms. They've already been diagnosed by a physician. They've already had their gene tested and confirmed. Okay, next slide. So every participant comes to Iowa City, the cultural center of the universe, right? Who doesn't want to come to Iowa City? Especially like in January, like a frozen wasteland. But for some people, it's a nice adventure. But we are only one site, and so this is the only study of its kind going in the whole world. And so, you know, we are happy to have this opportunity to serve families. So they do come to Iowa City, and so a lot of the program is organizing travel to get people to come here. Next slide. So it's a brain structure function study. So we use magnetic resonance imaging and MRI, and that allows us to take a picture of the brain. And we get volumes of specific regions. And the regions of the brain that we're interested in particular, we're interested in a lot of them. But certainly the caudate and the putamen are regions that we know are affected primarily by Huntington's, and we're looking at those regions. And we just look at simple things like the volume. So this picture gives us lots of, you know, quantitative measures. It's not just does the brain look abnormal, but that we measure things. And then we look at brain function by thinking skills. So we ask all the participants to do concentration, language, motor skills, and that's the brain function part. And then we're able to look at the two together. Next slide. So for the JHD program, it's the same thing. It's the research, everything's for research protocol, but they also get examination by clinicians. So once again, in the JHD protocol, these are kids who have already been diagnosed. So they will stay an extra day. And we have two teams. We have a team who is primarily for pediatric, so kids that are under 18. And then we also have kids who are JHD, meaning they were diagnosed before the age of 20, but now they may be over 20. And that constitutes Dr. Cam Holtz, who's a neurologist, and Dr. Moser, who's our neuropsychologist, and me. I'm not a child psychiatrist, so all of the children, even though all the research that I do is in children, clinically I see adults. So that's the additional piece that the JHD kids do. Okay, next slide. And then they come back to Iowa City. So in particular for the JHD protocol, we like to see them every year. Especially when you're very young, you know, your brain may be actually changing quite a bit. And so we like to repeat the protocol annually. Okay, next slide. So what are we looking for? What are we finding? That's, of course, an interesting question. Since we're so far along into these protocols, we can give you a kind of good sense of what it is that we're finding these days. So next slide. So in general, we're looking for the effects of that gene on brain development. So remember, any child, anybody who is born with the Huntington gene that's expanded, so they're called GE, the gene expanded, that gene is present from the moment they're born. And so that gene, which is important for brain development, is part of the normal development process. But in the children in which the gene is too long, it may affect brain development. So we're looking at that. We're hoping to understand what happens when the gene is expanded. Does it affect brain development? How? Do we see something early on? Do we see something later? Is there minor subtle changes that we see in childhood? And how is this manifest? So those are some of the things that we're asking is mostly how does this gene affect brain development? Next slide. So this is kind of a busy slide, but I'll walk through it with you. So remember I mentioned to you the pieces of the brain, the caudate and putamen, that's the region of the brain that's affected first and affected most severely in Huntington's. And so on the left side, you see a line going up, and that's the volume of the striatum. And so it goes from smaller as you go up, it gets bigger. So if we just look at the volume of the striatum, and you see these, what we call them glass brains, you can see the inside, that red part being the striatum. You can see that over time, I'm sorry, not over time, but as the CAG repeat increases, that the striatum doesn't grow as big. So the gene does affect the development of the striatum, and it affects it in a way that's dependent on how many repeats that you have. So for instance, if you have a repeat of 40 to 45, it does affect your striatum, and your striatum is not as big as somebody who doesn't have a gene expansion, but it's a pretty minor decrease. But as you continue to go up in the CAG repeat, you can see that the higher the repeat, the lower the striatum volume. And remember, that kind of makes sense because it goes hand in hand that the higher the repeat, the earlier the onset. So essentially, this is just saying that, you know, the repeats do affect how the brain develops. It affects the region of the brain that's most important for Huntington's, and it affects it in a way that it doesn't develop as much as it should. It's underdeveloped because of having the gene expansion. Okay, next slide. So what happens when your striatum doesn't develop properly? So there's another piece of the brain, it's called the cerebellum. It's right back here, it's right in the back part of your head. And that piece of the brain is connected to the striatum, and they develop together. And when one part is weak, the other one helps out. And so when the striatum is weak, when its development is stunted, so to speak, then the cerebellum helps out, or it compensates. And that's probably one reason why, even though the gene affects brain development, that the symptoms don't start right away. The symptoms start much later in life. And that could be because the cerebellum is kind of helping out the striatum. Next slide. So what we see when we look through the whole spectrum, we see that the cerebellum does indeed kind of help out with the striatum. So on the far left side of the graph, you see kind of a normal function, so a normal brain in which you have the striatum and the cerebellum talking to each other. But if the CAG repeat is 40 to 44, now those are people who will develop Huntington's disease, and their striatum is not fully developed. It's a little bit smaller. But the cerebellum can actually compensate, and it compensates very well, mostly because the striatum is only minorly affected. And that means for these people, they will go, you know, until the age of 40 or 50 before they develop Huntington's. And that could be because the cerebellum is actually keeping up with the compensation. Now, once the repeat gets longer, it's harder for the cerebellum. So in people who have a repeat from 45 to 59, now remember the striatum is even smaller because the development is affected even more so. And the cerebellum now has to work a lot harder, and it may not be able to compensate as well. And so these are people who will develop Huntington's sooner, okay? So they'll get it more like age 30 or so. And then to go to the far right, you'll see that when the CAG repeat is above 60, now the striatum is so small, its development is so interfered with, that the cerebellum becomes just actually larger than normal. It becomes enlarged. And at that point, things are really out of balance. And that's why the symptoms come on so much younger. So they come on below the age of 20. But this is how we view, the disease process, is that the Huntington's gene affects the development of the striatum. That development, that abnormality in development, is dependent on how many repeats you have. So it's only a mild abnormality if it's a CAG of 40, but it's a much larger abnormality if it's a CAG of 50 or 60. And then the reason why the disease doesn't come on, until many decades later is because the other part of the brain, the cerebellum, is able to compensate for the abnormal striatum. And depending on how abnormal the striatum is, the cerebellum is able to, keep things going normally until much later in life when then symptoms develop. Next slide. So this is just a picture of the lab. We have a lot of people involved. We have people who are involved in brain imaging. We have a lot of people who are involved in coordinating families and laying down the red carpet and doing all the assessments and being an awesome group to these families that come all the way to Iowa.
Beautiful information. This is all new information to me. And was this information that came to you through the brain imaging studies?
Yep.
It's phenomenal. My question is about the cerebellum, because what if the cerebellum is not compensating properly?
Yeah, if the cerebellum isn't compensating properly, then I think the disease comes on earlier, or it may be more severe.
The cerebellum, what if there are other factors involved in developmentally with the cerebellum?
Yeah, so if the cerebellum, I think that's the piece that's going to keep things going. And if it's under stress, if it itself is having its own problems from some other reason, then I think you can get more severe symptoms or early on. But these are relatively new ideas, by the way. The cerebellum in general has been considered the region of the brain that is just not affected by Huntington's, because we don't see it as being like abnormally affected. And some of that is because it's not. It's really the area of the brain that kind of keeps things rolling. And
I thought the cerebellum was part of the brain that was part of our ancestral origins.
Well, you're right about that. It's a kind of primitive part of the brain.
Yeah, that's it. That was it.
Yeah. Part of it, but actually, in the evolution between primates and humans, the biggest thing that happened was the brain got a lot bigger, and the two areas of the brain that got bigger with human brain evolution was the cerebellum and the frontal lobe. And this is the area of the brain in which all of the functions that make us human, so it's socialization, it's organization, it's, communication. And so this is a part of the brain that's really important. But yeah, so the cerebellum and the frontal lobe are the ones that got biggest in evolution.
We have a question regarding the study. Are travel costs covered for people who participate?
Yes, 100%. So families that come out pay nothing but their time, which is not, minor. We had a family come in today and they had because it's January, they had some issues with the weather and planes. But
yeah.
We cover all the costs. And so we're always very appreciative of anyone who wants to come and participate. And so all of the costs, so your travel costs, your meal costs, your hotel costs, food and parking, everything is taken care of. And then the participant is also provided some extra funds for their time and their participation, because we're grateful for that too.
So I have a question regarding the CAG. Have you found in any of the studies, or are you aware in any of the studies, where the CAG increases?
Over time?
Yeah, yes.
In the same person?
Yes.
So we know that increases in the brain. And so, the only way we can do these studies is look at the brain after someone has died, so that we can look at that tissue. So that area of the brain that I told you about, striatum, over time, the CAG in any one person with Huntington's will increase, so that you can get repeat lengths in the striatum, in the cells of the striatum, that are hundreds, even thousands long. So that happens over a person's lifetime. Now, when we do the gene testing, it's all from blood. So we don't, you know, we get the gene from your blood. And in the cells in your blood, that doesn't change over time. And so those are stable. So if I get your blood, the cells in your blood that I test the DNA, that's going to be about the same. So if I test you when you're 10, it's going to be the same as when I test you when you're 50. So it depends on the tissue. So like I said, the striatum is the area that we know is affected most. And so the CAG repeat gets very long. Interestingly enough, it does not get long in the cerebellum. In the cerebellum, it stays about the same length. It does not get long over time. So that's another piece that makes us think, maybe the cerebellum really is kind of the thing that's holding things together here.
And what determines how the tissue is affected?
Yeah, so that's
repeat, you know,
that's the question. We don't know that. Yeah, that area of the brain just seems to be the most vulnerable, you know, to this particular gene. We also know that there's probably other genetic factors that will determine that lengthening. So there are mechanisms whereby your cells try to repair. So if you, you know, get an elongation in your striatum and your CAG repeat, your cell's going to want to try to repair that. And so it's called DNA repair. And sometimes the genes that are involved in DNA repair, that may be a factor in when you develop it. So in other words, if you have genes that don't repair your DNA very well, you may get elongation quicker than somebody else. These are all just thoughts, but they're, you know, some of those DNA repair genes have been recently published to discuss, they appear to be factors for modifying when the age of onset is. So just because you have a CAG repeat of 40, like I said, you can actually have an age of onset in that range between, you know, 30 and 50. It's still a lot of range. There's still other factors. So one other factor may be your DNA repair genes and how efficient they are, and when you're going to get the disease. I hope that made sense.
Yeah, and then also another question that I've always tried to get answers for is passing the gene on to your children. You know, how does that work? And I mean, at what point does it increase?
Right. So for the most part, yeah. So for women who pass on the gene, the CAG repeat stays mostly the same. It doesn't really contract much. It doesn't really expand much. It's pretty similar. So you can, for the most part, if you've inherited a Huntington gene from your mother, it's usually about the same. In fathers, it can elongate. So it's called anticipation. And when that happens in the process of, you know, essentially of the making of the sperm, so that's a process that we don't understand. We don't understand why it elongates and why it's more likely in men than it is in women. Because there's other diseases that are very much like Huntington's. They're triplet repeat diseases. One of them is called myotonic dystrophy. I work in that disease too. And that one, it's the opposite of Huntington's, is that when mothers pass on that gene, it can elongate, but not with fathers. So these are things that we're just trying to figure out with that. But in Huntington's, the expansion, or what we call anticipation, is almost always when it comes from a father down.
Okay, so where are we at with the study? Are we going to be adding more subjects, or?
So for JHD, we're definitely adding subjects. So anybody who has been diagnosed prior to the age of 20 or 21, we're recruiting and seeing them annually. Currently, the kids at risk, we've currently closed further participation in that, enrollment in that. We hope that's temporary. We hope to open it up again once we get more funding. But this is a study we've been collecting patients since 2006, and so we're finalizing the data set and looking at it and doing some publications. But right now, the study that's recruiting is the JHD study.
Fantastic. Well, we hope to hear that you've received a new NIH grant very soon.
So do I.
And if we want to give to your research, how would we do that?
So the best way to do that is through our UI Foundation, so University of Iowa Foundation. And actually there's one contact person in particular, his name is David Maxey, M-A-X-E-Y. And he is the person in the foundation who governs donations to our hospital, in particular to Huntington's research. And so that would be the way to do it. Dr.
Nopoulos, thank you so much for your time. You are just phenomenal with research and people. You're such a great people person. We love you for that. And we love you for taking care of our JHDs and our HD kids. Sarah Jones has an announcement for us, and then we'll wrap this up. So again, Peggy, thank you.
Thank you.
Thank you. Yeah, that was an incredible talk. I mean, yeah, much. So that who is in Arizona area, or you can fly into Phoenix for this, that on February 20th, Kristen Powers will be in town, and she did the movie Twitch, and will be in Phoenix. The HDSA is bringing her in to the local chapter to do a talk at the Sontag auditorium. So you can reach us if you'd like information about how to reserve a space for that, but we want to make sure that people know they can always attend that if they'd like to.
Thank you, Sarah. Thank you so much. Thank you everyone for attending. Thank you all for listening. And thank you for joining us on Lunch with Docs with Dr. Peg Nopoulos. Have a great week, and we'll see you next month.